Leslie Vosshall. She is with Rockefeller University and has been studying the science of scent and odor and smell for a good many years, and now has been able to translate that science to working with mosquitoes and the reason why they are attracted to humans and why some folks get eaten alive more than others. She may touch on that, or if not, I'll probably be asking a question or two on that. I wanted to let you guys know that we, as everyone knows, we're very pleased that our caucus chairs, Congressman Rush Holt, Congressman Brian Bilbray, and Congressman Mike Castle are always our fine chairs, and we want to thank them for their support and their help with these endeavors. And I'm not going to take up much of your time. I want to bring up Dr. Vosshall and be ready to hear what she has to say, because I am one of those folks that get attacked by mosquitoes every summer.
Thanks so much. I'm really excited to come down and tell you about what we're doing. And I picked a May date because May is around the time when mosquitoes are done overwintering in the Northeast, and they start getting ready for the next season of biting and disease transmission. So I'll tell you today about some of the basic and applied research that I and my colleagues have been doing on mosquitoes. So why is this an important question? Mosquitoes are an annoyance to people living in the first world, so they ruin our barbecues, bite our children, make it difficult to go hunting, fishing, spend time outdoors. But in the developing world, mosquitoes kill. So mosquitoes are sort of known as the world's most dangerous animal by the large numbers of people that they kill, and they kill because, not because of the bites, but because of what's transmitted with the bites. So dengue fever is spread by Aedes species of mosquitoes, up to 100 million cases worldwide per year, and the number of deaths due to dengue hemorrhagic fever, it runs around 25,000 people per year. And as you see in the world map, it's sort of endemic in the Caribbean, Mexico, South America, regions of Africa, and Australia. So dengue is a disease that affects both developed world and developing world. And it's actually on the rise. So if you look at the number of cases of dengue hemorrhagic fever across time, it was quite low in the 50s when mosquito eradication programs were working very effectively. This is one of the few areas where, although science has been ascendant, mosquito control has suffered severe erosions, and with it, diseases are resurging. So you see this huge increase in Southeast Asia of dengue hemorrhagic fever cases. And the same thing is true in Central and South America. So cases are increasing from the 1960s through the present day, and this is not good. And just recently, from the State Department website, there are frequent dengue outbreaks that cause the State Department to issue travel warnings, either for South America or for Australia. And Aedes is not alone. Of course, there are many other species of mosquitoes that bite humans and transmit disease. Anopheles gambiae is the malaria vector. Aedes albopictus, the Asian tiger mosquito, is a yellow fever vector. And our own Culex pipiens is now spreading West Nile encephalitis, a virus that was not seen in this hemisphere until about 5 or 10 years ago. So this is a new disease that's been picked up by the southern house mosquito, Culex pipiens. And so all these variety of problems carried by different species of mosquitoes, either the parasitic animal that causes malaria or the viruses that cause West Nile, dengue, and yellow fever. And so malaria is also endemic and is a much bigger killer than any of the diseases I mentioned so far. So malaria is endemic throughout the southern hemisphere and affects sub-Saharan Africa particularly badly. So again, malaria is also a mosquito-borne disease. It infects 400 million people per year, has a much higher death rate, so kills between 800,000 and 1.5 million humans per year. And tragically, it's the very young who are the most vulnerable, and it's where the death rate is the highest. So the important thing that I'll try to convey today is that all of this is a product of this love relationship that mosquitoes have for humans, how their attraction to humans primarily through their sense of smell. So what are some strategies that scientists and people working in the field could could apply to deal with insect-borne diseases. You can sort of break it down into two parts. One would be that you could treat the humans. People have been working for about 50 years to make an effective malaria vaccine. The attempts so far have been not that encouraging. There's a new vaccine that has some potential. That would be one approach. The other for malaria is to develop new chemotherapeutic treatments, some of which now, new treatments are showing some promise. On the other hand, you might argue that the disease starts with a mosquito. So if you can stop the mosquito either from existing or from biting people, that's the most effective point at which to treat and prevent these infectious diseases. So things that were tried in the 50s, of course, is get rid of mosquitoes. So insecticides like DDT were incredibly effective at dropping down mosquito numbers, and that's why malaria and these other viral diseases and malaria were really knocked down in the 50s just by getting rid of mosquitoes. Another technique is to prevent the larval stage of mosquito that lives in water from growing either by putting poisons in or getting rid of standing water. Some modern technique that's being discussed by some colleagues is actually to genetically manipulate mosquitoes to make them stronger, so to make them resistant to malaria or dengue virus. These are very high-tech ideas that are currently being tried in the laboratory. There's a certain appeal to that. So mosquitoes exist in the food chain. They feed birds. They pollinate plants. And so if you could unleash a strain of mosquito that still bit humans but didn't transmit disease, that's an interesting and effective idea. And from our point of view, the thing that we're most interested in is just stopping the disease at the point of contact. So just interrupt biting behavior. Make mosquitoes not bite people, either by coming up with repellents that really work, that make mosquitoes not bite people, in conjunction with bed nets, which are a very low-tech tool but incredibly effective. And there's a lot of charities out there distributing bed nets under which people can sleep so they're not bitten by mosquitoes. So one of the problems of insecticide spraying, of course, is that many of the poisons sprayed from planes and trucks are also toxic to human nervous systems. So they kill mosquitoes just because mosquitoes are smaller and more susceptible. And many insects have developed resistance to insecticides. So this is the drawback of insecticides. They're toxic and they're failing in many parts of Africa. The state-of-the-art repellent, Deet, was developed by USDA scientists and the Army about 50 years ago. Nobody really knows how it works. And I'll go over in a minute the problems with Deet. And bed nets, incredibly effective, but also quite low-tech. The Army is also incredibly interesting. We have an Army meeting next door. The armed services are very interested in developing insect repellents because one of the great problems with going to war is that you send soldiers to disease-endemic regions where they're bitten by mosquitoes and they're infected with dengue and malaria. And this is a huge problem for armed services. So the Army uses the existing insect repellent Deet to come up with face paint. So the soldiers put face paint on and they also at the same time get a bit of insect repellency. But there are many problems with Deet, and I think both the public health sector and the armed services are very interested in coming up with new ways to repel biting insects. So what is this attraction that mosquitoes have for humans? So it comes from the need that female mosquitoes have for human blood. This is just a movie, a real-time movie of a female that's landed on someone's hand and is biting the person. So most insects on Earth feed on plants. There's only a few insects that require blood, and they require blood in order to mature their eggs. So they only go for a human blood meal when they need to ripen their eggs. And many of the species of mosquitoes I've mentioned bite almost exclusively humans, in particular the malaria mosquito Anopheles gambiae. So they have evolved this strong attraction to humans, and in doing so transmit malaria. And so this is the problem that's an interesting basic biological problem about how mosquitoes find humans, and also an incredibly important public health problem. So, and a nice demonstration of how basic this attraction is this funny experiment that Mark Clouden did in the 1970s. He grabbed a praying mantis, which normally mosquitoes ignore, and he manipulated it with his hands and covered the praying mantis with the scent of his hands, and then put the praying mantis into a cage with a bunch of hungry female mosquitoes. And they actually landed on the praying mantis and started feeding. So this is a demonstration of how important the human skin scent is for just triggering this feeding behavior. And so the central idea that it's how we smell that really causes mosquitoes to jump on and start feeding. So just to cartoon the process, mosquitoes first look for a host by the carbon dioxide and body odor that we give off, and then they feed and take their blood meal. And if they're infected with Plasmodium falciparum, then you'll get malaria. If they're infected with dengue virus, you'll get dengue. Then later on, the females, so only females bite, males don't bite, so the females will then develop their eggs, and then they'll go out and look for a site to lay their eggs. This is also based on the sense of smell, trying to find a place to lay eggs that has water in it, and then they lay their eggs. So both of these behaviors, biting and egg laying, are almost completely governed by the sense of smell. So it's an important thing where we could intervene in biting and in egg laying. So what is it about us that mosquitoes love? So first of all, body odor. So we all, although many of us wear deodorants, we still give off scents from our feet, from our underarms, and throughout our skin. And every time we exhale a big bolus of carbon dioxide. And together these scents are insanely attractive, so much that you can buy a product that mimics humans by putting out puffs of carbon dioxide and puffs of human body odor, synthetic body odor, and you can clear acres of land of mosquitoes. So now the question is, if we could figure out how mosquitoes are smelling us, could we then intervene and come up with high-tech insect repellents that would begin to solve these basic problems of mosquitoes finding humans? So I'll tell you two stories, how do insects smell carbon dioxide and how do insects smell body odor. So carbon dioxide sensation is really cool. If you record from the antenna of an insect, which is the insect nose, they can tell a difference from 300 parts per million carbon dioxide to 350 parts per million. So you can go from ambient CO2 up a little bit and the cell gets excited. You can go from ambient down a little bit and the cell gets excited. So they can tell a 50 part per million difference in carbon dioxide. They're very, very sensitive to carbon dioxide. So what is the protein in the antenna that allows them to do that? So Richard Axel, my postdoctoral mentor, and I, and also John Carlson at Yale, have been interested in carbon dioxide in insects. This is a movie of a fruit fly, which is a model insect that we use to study the biology of smell. This is a part of its brain, and this is the part of the brain that thinks about carbon dioxide. So it's a very small part of the insect brain, totally dedicated to smelling CO2. So when you exhale, this part of the fly's brain lights up. So what are the protein sensors that allow this to happen? We now know that two proteins called GR21A and GR63A are alone necessary to make a protein that can smell this gas. Really cool, just two proteins is all you need. And the demonstration of that biologically was that you can move the two proteins from this cell, which smells carbon dioxide, to this other cell, which does not smell carbon dioxide, and you can induce that cell to begin going crazy in the presence of carbon dioxide. And this is this experiment we published 2 years ago. So we're just recording from the antenna of an insect. If you move these two receptors, you get this big neural activation. So the demonstration that these two proteins alone are necessary and sufficient to induce responses. If we genetically get rid of one of these proteins, you get a fruit fly that completely fails to respond to carbon dioxide. So these bars just indicate how strong, so 80% of the flies respond strongly to carbon dioxide. If you make a mutant that doesn't have one of these carbon dioxide receptors, the response is 0. And that's a biological experiment that says if you could interrupt the function of this gene in a mosquito, either chemically or genetically, you'd have a mosquito that couldn't smell your breath, and that would be an important public health advance. And luckily for us, these are pictures of the sensory bits of Anopheles mosquitoes. And here, these colored spots indicate neurons, olfactory neurons in the mosquito sensory apparatus that are expressing carbon dioxide receptors. So we know that the same genes that are necessary for fruit flies to smell carbon dioxide are present in malaria mosquitoes. So that's a big advance. If we could figure out a way to jam those proteins, if there's just those two, there's a potential tool there to disrupt ability of mosquitoes to sense carbon dioxide. So what about body odor? I'm going to show you some cartoons about how we know how smell works in us, mice and humans. So we smell with these proteins that sit in our noses called odorant receptors, and they couple through a signaling process called G protein signaling. The same kind of proteins are used in worms that are used widely in biology, the C. elegans nematode. So we and nematodes and every other animal that's been looked at smells with G protein coupled receptors. And that's interesting because fruit flies don't. So fruit flies smell with these other proteins that are completely distinct from how humans smell and birds smell and dogs smell. And that's important because if we want to develop chemical blockers for these proteins, cartooned here, they should only work on insects and they should have no effect on humans, unlike insecticides, which just act as general neurotoxins. So these are the proteins that we're going to try to use to develop new insect repellents. And in particular, we're interested in one of these receptors. Most insects have about 100 different odorant receptors that they use to smell all the different things they care about. Medflies care about citrus crops, moths care about wheat crops, and mosquitoes care about our body odor. And they do this with a group of about 60 to 160 different receptors. If you look at the fruit fly, this is how the fruit fly smells, the things it likes to smell. And each of these little dots is a cell expressing is this receptor or this receptor, which governs how that cell smells. So this receptor makes these neurons be sensitive to benzaldehyde, the scent of bitter almond. And these cells expressing this receptor are activated by fruit. So depending on which of the 60 receptors you express, the neuron will be sensitive to different odors. But one of the receptors called OR83B, which is the core of our strategy, is expressed everywhere. So essentially every neuron expresses OR83B, and OR83B is widely found across the insect kingdom. So the medfly, the malaria mosquito, also the Aedes aegypti, yellow fever mosquito, the moth, all of these guys have this protein called OR83B widely expressed in all of their olfactory neurons. And that's important because we now know from a paper published last year that the specific receptors which bind to whatever smells the insect care about forms a complex with OR83B. And this protein complex together makes a smell receptor. If you get rid of this protein subunit, you make a fruit fly that smells nothing. A little bit like knocking out one of the CO2 receptor, we can make a fly that smells no CO2. If you knock out this subunit, you make a fly that essentially smells nothing. So the approach that we are taking, that we and others are interested in taking, is seeing if we can mess with this protein complex and jam the olfactory system of mosquitoes. And this is based on recent work from our lab and others investigating how DEET works. So DEET is something that you spray on yourself if you're camping. It's been in wide use. No one really has a consensus idea of how it works. Many people agree it does work. So it seems to repel mosquitoes. So if they get to the point where they land on your arm, they don't like how it tastes or feels, and they fly off. It can actually kill mosquitoes. If you put mosquitoes in a box with a lot of DEET, come back the next day, many of them will be dead. And there's also evidence that it acts as a distant. So if you spray yourself with DEET, mosquitoes will detect the DEET from a distance, either because it smells bad to mosquitoes, which some people favor, or our idea is that it actually blocks the proteins that allow mosquitoes to find us. And so we have some really good data for that. So if you record again from the malaria mosquito and ask, with this human body odor compound 1-octanthiol, this is electrical activity recorded from a mosquito antenna. That mosquitoes love this odor. This is the odor that you put into that backyard mosquito device to attract mosquitoes. So these lines indicate the mosquito organ is getting very, very excited by 1-octanthiol. If you do this in the presence of DEET, there's much, much less neural activity, suggesting that DEET is sort of shutting down or blunting the response of the mosquito to this odor. That and much more data leads us to propose this idea that what DEET is doing in part is jamming the ability of these proteins to work. So in the presence of DEET, the mosquito may fly toward you. As it gets closer, the DEET jams its smell system, and it's as if you've disappeared, right? So that the mosquito can't find you and you're not bitten. However, there's a lot of problems with DEET. So the commercial formulations have to be applied at really high concentrations. So in the business, we talk about molarity, how much chemical are you putting on your skin. 1 molar is a very high concentration. The Army puts on formulations as high, this is 25% DEET. The Army uses 98% DEET, so super, super high concentrations. You have to reapply this frequently. It has an unpleasant, greasy feel. It damages fabrics and plastics. And then most important for public health, it cannot be used safely on infants under 2 months of age. And this is one of the most sensitive populations for malaria. And there are these sporadic but persistent reports of neurological toxicity. Some people are sort of buzzing that part of Gulf War syndrome could be tied to the use of 98% DEET by the armed forces in Iraq, right? So all of these things are worrisome. So, and it's very old, so how far has technology come in every other sphere of human life since 1957? Cars, cell phones, cancer drugs, insect repellents have not budged since 1957. So we want to use what we know about the insect sense of smell to develop new blockers of this class of proteins that work on this OR83B protein, which is essential for all insect smell. And we do this by using modern screening techniques that are in use by the pharmaceutical industry. So we have our protein targets, so we know which protein we want to inhibit. We can make cell lines, and we can reconstitute mosquito smell in a tissue culture cell. So you get a tissue culture cell, you load it up with mosquito odorant receptors, you apply human body odor, and the cells will glow. They'll fluoresce in the presence of the body odor. And we just look for a chemical that blocks that glow. So it's a very fast way where you can screen through candidate compounds, chemicals that will be the next generation of DEET. Here's an example of one of these experiments. Blue, this is it's called a 384 well plate, so we can look at 384 different samples at the same time. So in blue are samples that have not been activated by the body odor components. These are wells that have only gotten the body odor, so they're glowing yellow-green. And then the rest of the plate is individual wells that have the body odor plus a chemical. And of course, this is our favorite chemical here because this is something that completely blocks the ability of this smell protein to function. And so what we're in the process of doing is translating this from the laboratory to ask, do these compounds, when applied in the laboratory, block the ability of fruit flies to smell and the ability of mosquitoes to smell? With the idea that by blocking the basic mechanisms of insect olfaction, we can potentially bring to market something that will knock DEET out of its place as the #1 insect repellent in the world. And because the proteins that we use are insect-specific, they should not have any off-target effects on proteins in humans. So that's sort of our dream in the field as a whole, to try to come up with new repellents based on what we know about insect olfaction. And all of this stuff has been made possible initially by the Bill and Melinda Gates Foundation, who are very interested in coming up with new technologies to address persistent problems in public health, by the National Institutes of Health, and then recently by the Howard Hughes Medical Institute, which has made the mosquito work in my laboratory possible. Thank you. It's such a persistent, pervasive anecdote that I think it must be true, and it's something that my lab is really interested in. So no matter where I go, whether I'm in the airport or at a family reunion or meeting with students, everybody reports this phenomenon that mom is always bitten, mom is always bitten, dad is never bitten. The 4 children, 2 are bitten, 2 are never bitten. It almost looks like a genetically passed down trait to be bitten or not bitten. We know a little bit about what chemicals make mosquitoes attracted to us, so it must be that the people who are always bitten are broadcasting something incredibly attractive that brings in mosquitoes. Alternatively, the people who are not bitten are broadcasting a natural repellent, right? I think that's very testable. It's extremely testable. And now that with Hughes money, we have mosquitoes set up in the lab, and because we do a lot of human subjects research, my dream is to start recruiting people who report that they're never bitten or always bitten, get them to wear some t-shirts, we cut up the t-shirts and we test, is it really true? And then we ask, why is it true? So I think that's another important observation that could be a route to something useful and important.
Since I am, I feel like I've tested everything out on the market, and recently, you said not much has changed, but in the past couple of years, or at least 10 years or so, Skin So Soft, the Avon product, seems to work well. And we were talking about this earlier, the citronella candles that we try to put out to block them. It's when we're outside, besides loading down with DEET or any other kind of thing, is in the mosquito lamp itself. Is there anything that we can do to clear our yard?
So I think that the major repellents that are out there are DEET is the raw ingredient, Skin So Soft, which is a new compound, and then in Europe people use Bay Repel, picaridin. All those have more or less the same efficacy. Some work better for some people. There's nothing that's out of the park better than any of those. Then there are the natural products, which many people swear by. There's no good laboratory evidence that citronella or any of the herbals are nearly as effective as DEET. So this has been a problem in humans for millennia, right? And so we've dealt with it by burning herbs or setting fires, so that there's a rich folklore of what works and what doesn't. Our goal is to get past the folklore and come up with something that's 100 times better.
What are in those candles though? Is it DEET in the candles? You know how they have the outdoor candles that you light that's supposed to repel?
I wish I could answer that question. So many of them have
smell.
some of them have citronella, which does have a very strong smell, which may confuse the insect because it's flying into a cloud of citronella. Some of those products have pyrethroids, which are insecticides, which confuse insects, can knock them down. I don't know whether they have DEET, so I haven't looked at those.
And are those, with the evidence that you've shown by using DEET, it can have neurological effects, do these candles emit the kind of same things? Because, obviously you're breathing those things in.
Yeah, so I don't know. Obviously, the doses that you would experience, if those products do have DEET in them, the actual dose that would reach you is much lower than if you apply it topically. Then there's really good evidence that when you apply DEET to your skin, a lot of it actually goes through your skin and goes into your bloodstream. So that's a pretty effective route to take up DEET. The candles are probably less effective.
Thank you.
I wondered how you, focused your target search for assays, and secondly, the blue one, the one that seems to be effective, is it similar to DEET?
Oh, good question. So we really did it like Big Pharma does it. So we have no preconceptions. We have a target. We have a cell-based assay where we can reconstitute mosquito smell in a dish. And then we screen a sort of a random chemical library. So it's a combinatorial chemistry library that can be used for cancer screening or hepatitis screening. So it's not biased toward anything. And what I didn't say, thank you for asking, is that we have about 10 compounds that we're really interested in from our initial family of 100,000 that we screen. And none of those 10 looks anything like DEET, which we're really happy about because many of the efforts in the past 50 years have looked at DEET and tried to tweak it to make it better, in my opinion, with marginal efficacy. So that hasn't worked so well. So the unbiased approach, I hope, will be a different way to do things.
What are the toxic properties of those 10 compounds that you find favorable? Are there some better than others, or?
We haven't gotten to toxicology yet. So what we know about DEET is that if you ask how selective is DEET for insect odorant receptors, the answer is not selective at all. So DEET will block human ion channels, the ion channels that we use to see, the ion channels that we use to feel pain. So DEET has very little specificity for the insect odorant receptors. So we're looking for something that will only act very specifically just on insect smell. And then down the road, we have to look at toxicology. So we want to make sure that they're just generally safe. But we're not there yet.
If we had an effective repellent that keeps the mosquito from biting us, that doesn't take away the mosquito's biological imperative to get some blood and keep on going. So are we looking to just send them to the next neighborhood, or have the mosquito die because they can't get blood? I mean, fighting Mother Nature this way seems like it usually loses.
So in the beginning, malaria is a really complicated problem, and I don't think any one of the solutions that are being pursued is the vaccine that that's currently being tested, made by GSK, has about 50% efficacy. The chemotherapy works in some places, doesn't work in others, depending on resistance. The repellents, I agree with you. I think it's unlikely that we'll get a repellent that will prevent 100% of the bites. I think the goal is to kind of integrate these approaches and have, if you could have the mosquito bite you once, but then not bite you again, then you would eliminate transmission, right? So the goal is to interrupt transmission. We don't have to prevent 100% of the bites. We just have to reduce the number of bites, and that's what bed nets do. So bed nets they work well enough that they actually reduce the number of bites and thereby reduce transmission. So
The first bite gives you the disease, and then they come back and pick it up again and give it to somebody else.
exactly. Or if you're infected and the mosquito isn't, and the mosquito bites you, if the mosquito then doesn't bite anybody else, that would be a good thing. So.
Are those notions of changing the genetics of the mosquito, is that going along any far along? And if so, how do you apply that to the mosquitoes?
So I think the biggest success story so far came from Australia last year. So Scott O'Neill's group used a natural means to introduce a bacteria called Wolbachia into Aedes aegypti. And this slightly reduces the lifespan of the mosquito. So it's a young and healthy female mosquito, and it can breed and it can bite. But because dengue virus it takes a very long time for dengue to mature before it's ready to transmit. Those mosquitoes are probably unable to transmit dengue. And because they're just carrying a natural bacteria, and the bacteria has this genetic imperative that it transmits itself, so unless you have the Wolbachia, you don't live. It's a way to propagate this idea through all of the Aedes aegypti in Australia. So it's kind of like a Trojan horse. So they've injected a Trojan horse into Aedes, makes them live 5 days less than normal, so they die at 12 days rather than 17 days, and they're probably not going to be able to transmit dengue, and they'll take over. So that's an idea that the Gates Foundation funded that I think will work. And because it's sort of natural, you're not really putting genes in, I think the public will accept it. The other ideas are a little harder because then you're putting genes in, and the public still is very wary. I think just because of scientific literacy, they're wary of genetic payloads. And those mosquitoes that are carrying transgenic technologies may be a little bit less healthy. And so there's the problem. You might have a great idea and you send the transgenic mosquitoes out and they're really strong. It's a really great idea, but then they don't breed and they don't take over the population, right? So I think the case in Australia, I think has a good chance of working for dengue, which would be a wonderful thing for the Gates Foundation, an idea that they proposed actually has real-world applications.
Are the other repellents you said were used in other parts of the world, are they, like you said, tweaking the Deets chemical? Is that where they come from?
Bay Repel has some structural similarity to Deet. It's a little hard to find out how they made Bay Repel because it was made inside a company. Yep. It has about the same baseline efficacy, so it's not really statistically better than Deet, but it's different.
Is it less toxic? Or?
It's pretty new, so I don't. I think of course the companies who make these things have to put it through basic toxicology. So of course we have to assume it's safe. Deet continues to be registered as safe, of course, and it's in wide use. It's just there. So over the last 50 years, there are these sporadic but persistent reports of neurological problems. I think we'd have to wait to see what happens with Picard and Bay Repel, like every other product that's launched. You have to wait. Sorry?
You can't buy those here yet.
Picardin is a little bit harder to find. It is. I have seen it in a few regions around the country. Yep. It's not as easy to find as Deet.
Yeah, I've got a bottle of something besides Deet. I think it's that, Picardin, a cutter product or something.
Yep.
From Walmart.
You mentioned earlier that they use some pesticides in the water where the mosquitoes will lay their eggs. Are those environmentally dangerous? Has that been tested, or is it?
Right, so in the 50s, when it was really the heyday of eradicating malaria by eradicating mosquitoes, quite successful. So DDT to kill the adults, and then usually kerosene put on the water to suffocate the larvae. So yes, both of those are environmentally pretty disastrous. DDT is now becoming back into use in Africa, but spraying inside homes, not spraying environmentally.
Is that
So
okay to spray
I think-
inside?
So this is called indoor residual spraying, and I think it's a risk-benefit ratio. So the risks of contracting malaria, I think, far outweigh the risks of spraying DDT in your house. So those old technologies are being brought back in endemic areas in Africa, just because no other new technologies have come along. So they've gone back to the old technologies.
Was there any environmental consequences from eradicating mosquito populations? I mean, does it affect the food chain somewhere beyond that?
I guess most ecologists would worry about complete eradication. So in fact, they haven't been eradicated. I mean, eradication didn't work, right? They came back. So there's only been a few cases of insect eradication. Screwworm was eradicated in the U.S. by the agricultural agencies, but yeah, eradication is for a persistent insect and a smart insect that's co-evolved with humans. I think is tough to do. Probably not wise.
And the only thing that makes bed netting safe or so effective is that for a prolonged period of time, the person is stationary and therefore much bigger.
So the reason that bed nets are effective is that Anopheles gambi, which transmits malaria, bites in general at night. So they have this interesting biology where they just wait until we're asleep, exactly as you said. And the bed nets work as a physical barrier, so they can't get to you, and also because they're coated with pyrethroid insecticides. And the new bed nets are very good. They're dipped in insecticide and they'll last for 3 to 5 years. So in areas where you can't go to the Walmart and get a new bed net, that's a really good technology. And it exploits the biology that those mosquitoes wait on the walls of your house essentially until you're asleep. Very sneaky.
The 10 compounds that you identified, what are you going to do with them next? I don't know if you maybe sort of touch on it, but what are the next steps?
So the next steps is very similar to what Big Pharma does. We have what's called a hit or a lead compound of interest. So we want to make sure that it's not generally toxic. So we want to test it against many other cell-based assays just to make sure that it doesn't poison lots of other cellular processes. Then we want to test it on fruit flies. So we can puff odors at a fruit fly and record electrical activity in the antenna. We want to ask, do our compounds block the responses to odors, just like Deet does. So Deet will block responses to odors. Do our compounds have the same effect? And the answer is they do. So some of our compounds do work on real animals. And then the next step is we do that in mosquitoes, recording electrical activity from antennae. But the real test is behavioral. So if you put mosquitoes into a big box with my hand at the end of it, and this hand is coated with the compound and this one isn't, will they not bite this hand? Is the test, and I hope that we'll do that over the summer, so we'll have real evidence as to whether this idea that came from the basic research laboratory side of things has some plausible efficacy. So check in at the end of the summer.
After you did that.
Yeah. In the meantime, stay indoors.
I've heard there are other ways that mosquitoes can find you. They also see infrared. Is that right?
Yeah, mosquitoes they're sensitive to lots of things. So the females are sensitive to water, obviously, for egg laying, and they are sensitive to heat, which is infrared energy. So many insects can see ultraviolet light, but no one has really done the rigorous test of which of these is the most important. So is it our body heat, is it our carbon dioxide, or is it the body odor? So we're trying to do that this year by making a mosquito genetically that can't smell carbon dioxide or that can't smell body odor, just by the genes I talked about. So we'll be able to answer your question. If you get rid of all of that and you just have heat, will they still be able to find us? I think that they won't. I think that the smell is a really important part of it. Thank you very much.